Now we are looking in terms of further applications of the polymerase chain reaction. You understand polymerase chain reaction had been one of the sort of a tools which is used in recombinant DNA technology or biotechnology. Now PCR has got a very important application in early disease diagnosis. Early detection of the disease as well as a diagnosis of the sort of a disease. Like which are all the common diseases which can be detected by means of the PCR. We have, it is useful for the detection of AIDS disease. AIDS disease. Again, we have a very important disease which is termed as hepatitis, which is caused by the C virus. Hepatitis C virus, early diagnosis of hepatitis C virus. Then it is also important for the detection of TB, tuberculosis. It can detect genetic variants. In any population it can detect genetic variations. PCR is also useful for disease diagnosis. If you want to exactly say whether a disease is latent or it is progressing or whether it is an epidemic disease or else whether the disease is epiphytotic, that is restricted to a small sort of a geographical region. For detection of all these particular diseases, primarily a PCR comes as a very important sort of an aid. So, infections which are caused by epidemics leading to infections can also be detected by means of PCR. What sort of an infections are spread in the human population because of an epidemic. And very important application of PCR maybe in the future, as of now, is also in neonates, that is in newborn babies in the first week of their life. We can very easily clearly do early disease diagnosis of a baby which is just born, and thereby we understand PCR has got lot of applications in improving healthcare.